I really want to do a review on this extraction, isolation, purification, and quantification or quantitative estimation of proteins. Proteins are quite numerous. A particular cell can house, hundreds and hundreds of protein in just one single cell, and then it becomes, very, very necessary for you to know how you're going to isolate or extract this particular protein in the multitude of other proteins, owing to the fact that proteins control virtually everything in the life of an organism. That is why most of the phenotypic expressions of genes that are seen are all as a result of translation of our DNA down to the protein. So when we say that the DNA contains the genetic information of an organism, we are indirectly saying that all the proteins that the organism is expressing is coming from the DNA. Now, these proteins, they vary so much to hard surfaces like the nail, the hair, very solid materials, to solutions like enzymes. Okay, and we know that proteins have different functions. We know that they have different properties. Now, these properties are what you're going to be looking out for whenever you want to isolate a protein. You must know the characteristics of that particular protein has, that will enable it come out of solution while other forms or other types of proteins remain. Okay, so we are going to go through this study one more time. Now, we are saying in methods of isolating proteins, isolation and purification of a single protein from cells containing a mixture of thousands of unrelated proteins can be achieved because of the remarkable variation in their physical and chemical properties. Okay, now that's what I just explained. The physical properties and the chemical properties of proteins vary from one protein to the other. So for you to actually, isolate this protein, you must know these characteristics of these properties we are talking about. Now, the characteristics unique to each protein comprise of the following. Proteins are different in their amino acid composition. The number of amino acids differ from one protein to the other. The sequence of the amino acids. When we talk about sequence, we are talking about the arrangement of the amino acids in the protein. Okay, whether there is aniline, there is a glutamine, there is a proline, or whether the proline comes first, aniline. The more the proteins change in their sequence, that what actually makes them different, and their properties will also vary. Now, the subunit structure is also very important. The size of the protein can also help in the isolation of such protein. The shape can help, especially in enzymes. We can make use of their shape, the shape of their active sites, the kind of substrates they are attached to. We can make use of all these things when we are talking about the characteristics that we are going to use to isolate them. We talk about their net charge. We talk about the isoelectric point or isoelectric pH. We talk about the solubility of such proteins, the heat stability. We also talk about the hydrophobicity of the proteins, the type of ligand stroke metals binding properties, are also put into place. Even the post-translational modifications, how is it they are modified after they have been translated from messenger RNA to proteins. All these can be exploited in extracting such proteins. Now, always take note of these characteristics. because an examiner might be interested in actually asking you list or describe the methods or the characteristics of proteins that can enable them in the isolation of proteins. So you must know each and every of these properties, what each of them do, what method could be employed when you are considering a particular characteristics. We see some of these things by the time we come to the end of this revision. Now, in isolation of proteins, to isolate a protein, the site of location must be considered, whether the protein is of the plant, animal, or microorganism origin. Okay. In other words, whenever we want to isolate a protein, we must know the source of the protein. Yes, whether it is going to be from plant, like if I'm asked to isolate papain, and I'm looking at animal um, models, it can come because papain is a plant protein. You get it from pawpaw plant. Okay, so you must know whether it is from plant or whether it is from animal, and you know the function of such proteins. Now, selection of the tissues. So, like I said, you must be able to select the tissue or the cell from where you're going to do your isolation from. For instance, when you're considering isolating proteins from the blood, okay, you must first of all know the site of the tissue. For instance, you want to carry out electrophoresis of the hemoglobin. You're doing genotyping of the hemoglobin. First of all, you must make the blood to release this hemoglobin, okay? And that is why you put this blood that you have gotten from an individual into a micro well and you add water. So the water will cause the blood to burst open. The cell of the red blood cell bursts open, releasing this hemoglobin for the study you are about to do. Do you understand? So you must know the place and the way to concentrate it. For instance, you want to work on with microorganisms, especially bacterial cell. You know, when you get the broth, you are expected to centrifuge it. And in centrifuging at about 16,000 G, you will see there will be a separation. The cells will go down to the bottom of your tube, your centrifuge tube, and then you can then resuspend it in the buffer and then use it for your studies. Okay, now let's look at tissue and cell disruption. The proteins are contained inside the cell or inside the tissue. So for you to actually get out your proteins, you must first of all burst it open. Okay, so there are in isolating proteins, the tissue need to be disrupted, and there are several methods of disrupting the tissue, which depends on the tissue source, the toughness, and the nature of the protein that you're obtaining. Now, if you want to get a protein from a plant source, you're not going to apply the same method to the one you're getting from animal source, owing to the fact that proteins, the animal cell are tougher. The membranes are tougher because they contain cellulosic cell wall. Okay, they have cell walls that are made of cellulose, so it is tougher. You must use a tougher method to handle such than when you are just getting from the blood cell or the membrane or the ordinary animal cell. Okay, so but now when you disrupt the membrane, there are two things that you must have in mind. One is to maintain the buffer condition. So you need a buffer that will have to maintain the pH of the place so that you don't bring out the protein from the cell or from its position and it gets denatured immediately. So a buffer is necessary. Then also an antioxidant is also necessary, because during the process of your extraction, there might be possibility of generating some reactive species okay, or yeah, oxygen reactive species. So the use of antioxidants can also be effective, like beta um, catetanol, the metacatetanol, that it helps in maintaining the oxidant status of the protein. And then what we just have here is a little diagram, showing us what happens during breaking of the cell or the tissue. Okay, for instance, you see the tissue or piece of the tissue that is in solution. Okay, the first thing you do is you use any method, which is either gentle mechanical procedures, something like homogenizer. You can use it to blend, but when you're blending, you have to take care that heat is not generated in case your protein is easily destroyed by heat. So most of the times when we are blending, we add very cold buffer so that we don't generate heat that will denature our protein. So some of these methods are what we use, but the most important thing is that the cells are broken open. Now, let's look at the method that we use in disrupting this membrane. Number one, you have the notes, so I'm not going to be wasting much of our time. You have the vigorous method, and in this vigorous method, we make use of what we call, one of the method there is a French press. We have a link in our notes in case you want to see the French press. You can always copy that link on YouTube. Now, what it does is that the machine presses down on the membrane that we want to extract the protein from. Okay, now when it presses it down, you know it will kind of release itself by force, thereby tearing the cells apart, and they get ruptured. Okay, so it finds a way of using the shell threads to disrupt the membranes of the cell. We say there are some disadvantages to this, which you can always see in the note. These disadvantages, the sample have to actually be very small. Procuring the equipment is quite expensive, and then you still have to do some homogenizing and all that. So you see that sometimes this method might end up destroying your protein. Okay, so number two is sonication, and sonication has to do with using very high frequency, in ultrasound to actually break um, um, the cells. Although this actually is also a very good method, because you use sound energy now, it does not generate as much heat as we generate in others. You can actually use a tissue miser, a polytron, which is a tissue miser. A tissue miser kind of acts like a blender, but the speed is quite enormous, so you can't avoid generation of heat and even mechanical disruption of the proteins when you produce them. You equally have what we call bead shakers. All these can be employed when you're making use of a very tough way to disrupt, a vigorous method to disrupt your proteins. Now we have a moderate method. Okay, a moderate method, most of the times we make use of blender, we grind with abrasive, you use mortar and so on. So you do it manually most of the times. Is a moderate method of of um, breaking open the cells. Now the third one is a gentle disruption. In gentle disruption, we normally apply them when cell walls are not available. So we make use of that in animal cells most at times, like in the blood cells, which I talked about some minutes ago. you just add water. And when you add water, you have changed the osmotic pressure of the cell. And when you do that, the water rushes inside the cell and break it open. Okay, whenever you make the environment of the cell to be hypotonic, water tries to move from the environment into the cell and the cell will burst open, and that is the one we call making use of the osmotic shock. Now you can use chemical solubilization. You use agents that can solubilize the cell membrane, something like detergents such as Triton X100, sodium chlorate. You can make use of isozymes. You can make use of lipases. Okay, so all these things can act for chemical solubilizers. And we talked about homogenizers, which you just use to grind whatever you want to isolate the protein from. But mind you that when you are grinding, like I've mentioned before, be careful of mechanical damage on the protein. Be careful of temperature, okay? Because sometimes you might want to use all this method. At the end of the day, the protein is denatured before the experiment takes place. And when the protein is denatured before the experiment takes place, then it will definitely affect the result of your experiment. Okay, now when we have finished grinding, the next thing is separating this. So the first thing we do is separating the large particles. So what methods do we use in separating the large particles? One, we can use a cheesecloth to remove the bigger particles. Okay, down here you can use that cloth they use in sieving pap or akam. That's cheesecloth. So you can use it, remove the big particles from, because when you burst the cells, every protein in that particular cell goes into solution. Okay, so as the liquid is draining from the cheesecloth, the proteins are also moving out through it. Okay, now you can screen by calibrated wire screen, okay, which you can use. It's still like a sieve, which you can use to remove broken and non-broken eukaryotic cell walls. Okay, because definitely this method was not going to be selecting the cells one after the other to break them down. Definitely there will be cells that are not broken, so you need to remove those ones. You can then filter. Number 3, we say filter. Okay, and then all these things is what you do to remove large particles. Sometimes you can centrifuge at a very lower speed, and when you centrifuge at a very low speed, cell debris will actually be removed from the cells. Okay, now in fact most of the thing that is commonly used is centrifuge. Once you filter, the next thing is centrifuge, provided you know the places that the particular location of your cell, of your proteins. Okay, it will actually help. So we always make use of differential centrifugation most at the times. And what is differential centrifugation? It means you're centrifuging at different speed, so that you can remove the sediment, the filtrate that you don't want. For instance, in bacteria, you try to isolate proteins from bacterial cell, and you use the osmotic shock. Maybe you now use salt of high concentration, or you use solutions that are very hypotonic in nature, so that they can flow into the cell and burst it open. Now, when you gather, the first thing you do is you centrifuge at say 1000 G for 5 minutes. And when you do that, it's expected that all the cells that are not tampered with will settle at the base of the tube. So you will decant and you now centrifuge again. This time around, you will increase the speed of the centrifuge and you centrifuge at 10 minutes. Now, when you do that, some other heavier components of the cell, such as if it's plant cells, such as the chlorophyll, the chloroplast, sorry, the cell debris, the nucleus, can go. Okay. Now, when you increase to 15,000 G for 20 minutes, the mitochondria can be separated at this point. So if I'm working with mitochondrial proteins, it means that when I get my cell and bust it open, I will kind of do a differential centrifugation, and when I get to 15,000 G and I centrifuge for 20 minutes, everything I'm supposed to get should be higher amount of the mitochondria. I don't know whether I'm making sense. Now, if you increase again to 30 G for 30 minutes, you can see that small organelles will be gotten and so forth, and just like that till you get down to the membrane sheet. So this method of separating by increasing the time and the speed of the centrifuge is actually known as differential centrifugation. Now, when we've gotten this protein, everything is in liquid. So what are we going to do? Are we going to continue with the work? No. We need to bring out the proteins from the liquid. Remember, the protein is soluble. So how do we do it? Well, we precipitate the protein. Now, in precipitating the protein, we put into account the solubility of this protein. Okay? That's why we said one of the things you watch out when you want to get out a protein is that you have to talk about some of its characteristics. Now, we said proteins usually carry charges due to the hydrophilic amino acid and the terminal amines and carboxylic acid. You know that proteins, amino acids are charged. So when they are connected in proteins, the protein can equally be charged. Now, because they have these hydrophobic amino acids on their surfaces that attract water molecules interacting with them, proteins are soluble in solution. So the solubility of a protein is a function of their ionic strength and pH of the solution. Okay, so for protein to be soluble, it's a function of two things: the number of charges it carries, which have to contribute to the ionic strength, and then the pH, whether it is in the acidic form or in the basic form. Now, proteins have isoelectric points at which the charges of their amino acid side chain balances each other. At this point or in this point, the protein may lose its solubility and precipitate out of solution. Now, when we talk about isoelectric points, like we are saying that it is a point where the net charge of the protein is 0. Okay, if it is a positive charge protein, okay, you can begin to change the pH, okay, to a point where the charges on the protein balances each other. Balances means the number of positive charges and the number of negative charges are equal. Okay, at this point, the protein might lose its solubility and come out of proteins. So we might make use of such things. But let's look at another thing we consider when we look at the solubility of proteins. Now, proteins have a way, if you have a way of making water not to be available for the protein can easily come out of solution. Okay, and how do you make water not be available? We can put an ionic compound that are equally soluble in water that can compete with the solubility of the proteins. Okay, so we say the solubility of protein is a function of its ionic strength, which we have said. At low salt concentration, the presence of salt stabilizes various charges on the protein and thus attracting proteins into solution and hence the solubility of the protein. And this is what we call sorting in. In other words, when I have proteins in solution or my protein don't want to dissolve, maybe because the environment is not quite ionic, I can increase the ionic nature of the solution, and when I increase it a little, the protein can go into solution very well. Okay, and that is what we call salting in. You're adding little concentration of salt to enable the protein go into solution. That is salting in. Now, but as you continue to increase this ionic strength of the solution, that is, you continue to add this salt, like I said before, more of the water is being dissolved. The salt is being dissolved in the water, making less and less water available for protein to dissolve, and that protein will come out. And this is what we call salting out. So in salting out, you will increase the concentration of the salt and you add it little by little. Now, this process of salting out is often used in precipitation of protein. In fact, you cannot do isolation without salting out. And the most common salt that we use in salting out is ammonium sulfate. And why is ammonium sulfate used? Ammonium sulfate used because it has an unusual solubility in very cold buffer. When the buffer is cold, most solutes don't dissolve when the temperature is at the extreme, very cold. Do you understand? But ammonium sulfate dissolves perfectly well even at -4 degrees. So we normally use it, in salting out protein. Now, sorting out is the first step. After you have removed this, you have done your filtration, you have removed all the debris, and you have protein in solution. In bringing it out, the first step is your ammonium sulfate precipitation. Okay, like I said, it is adding ammonium sulfate little by little, and the reason why we add it little by little is so that you target a particular protein at a point. If you pour it, for instance, if you pour salt in water, okay, you just carry one teaspoon of salt, put in a cup of water, you will notice that the base will have very high concentration of salt, while the surface will have low concentration of salt, and that is why you begin to stir. Okay, now, but if you begin to add salt little by little, you stir, little by little you stir, you see you have uniform concentration across the solution. So if you pour salt, that place that is highly concentrated, we might precipitate proteins that you don't need because of the high concentration at the base. So we normally add it little by little to avoid what we call co-precipitation. Co-precipitation, that is precipitating the proteins that are not your protein of interest because of variation in the concentration of ammonium sulfate. Okay. It's not only ammonium sulfate you can use some other organic compound, something like polyethyl glycol can be used as PEG, can be used. You can also use something like ethanol, acetone, they can precipitate protein. We can also, like I said before, adjust the pH to precipitate, and so on and so forth. So when you go through the notes, you will get some more insights. Now, in using ammonium sulfate to precipitate proteins, we make use of what we call the ammonium sulfate precipitation table. It gives you the particular amount of ammonium sulfate to be added so that you can achieve a particular percent of precipitation. Okay, so proteins are precipitated from aqueous solution by dissolving ammonium sulfate into the protein solution. The following tables show the weight of ammonium sulfate to be added to 1 liter of solution to produce a desired change in concentration. That is what we call percentage saturation of ammonium sulfate. Now, precipitation of proteins is conventionally carried out at 0 degrees to avoid possible denaturation of proteins, although percentage saturation value showed in the table is based at a value of 25 degrees described by Green and Hughes. Okay, now what are we saying? Normally, like I said, because of our proteins are denatured by heat, we normally work at cold temperatures, and ammonium sulfate dissolve very well in cold cold environment. Okay, now the values we have here says is the gram of ammonium sulfate that you dissolve in 1000 mils. That is 1 liter, 1000 mils of your protein. In other words, what am I saying? To get 10%, now when you look at the horizontal line, the horizontal line that is bold, where you have 10, it didn't start from 0, it started from 10, 15, 20, and so on. Those ones are the percentages of the ammonium sulfate you want to carry out. Okay, then the vertical one, that you have 0, 10, 15, and others one, are the percentages of ammonium sulfate that are already in the protein concentration. So when I have not added ammonium sulfate and I want to achieve a 30% ammonium sulfate precipitation, what I will do is that I will get, say, 100 mils. And if I must work with 100 mils, it means I'm not working with 1000 mils again. So I have to scale down. Okay, so since I'm looking at 30%, remember I will work with 0% because there is no 0%, no ammonium sulfate in the solution yet. So I will add 17.6 because 17.6 should be equivalent to 100 grams. 176 to 1000 mils, then 17.6 should be to 100 mils, or 1.7 to 10 mils. But now, in the experiment, we don't actually know at what ammonium sulfate percent that we precipitate our protein of interest. Okay? So what we do, or what is advisable for us to do, is to get different test tubes, say 1 to 10 or 1 to 20, so that we can accommodate all these percentages. Say 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. So we get 20 tubes and we pour 10 mils of our protein solution. Now, when we add 10 mils of our protein solution to the first tube, we will carry out a 10% ammonium sulfate, and we are considering the column that has 0. Okay, now when you consider the column that has 0, it means for my 10 mils I should add 0.56 grams of ammonium sulfate. For my 15%, I will add 0.84 grams. You have to work it out. For my 25% ammonium sulfate precipitation, I will have 1.44, down to when I will have 100%, I will add 7.67. Okay, this is to 10 mils of the solution. So I will shake it. Remember, you're working in cold environment. You're working in cold environment. Now, in this cold environment you're working, you will allow this, put it in the freezer or in the fridge for 24 hours, and then you will centrifuge. After centrifuging, it's expected that the proteins that you will get will settle at the base for each ammonium sulfate precipitation you've done. But your protein of interest might not be present in all. So what you will do is that after centrifuging, you will decant and reconstitute with your buffer. And when you reconstitute with your buffer, what you will do now, you begin to test each tube for the presence of the amino acid, for the presence of the protein of interest. So maybe the presence of protein of interest is in this place that have the one that has 50%. What you have just succeeded in doing is to say that the saturation of your protein of interest will be at 50%. Okay, so what you are indirectly saying is that if you get 1 liter of our protein solution, that's the raw protein solution or the crude protein solution, that you're going to add 313 grams of ammonium sulfate little by little, and you will stir it, put it in the fridge for it to cool, and you centrifuge. That whatever protein you will get will be your protein of interest, because your protein of interest precipitates at 50% ammonium sulfate precipitation. Okay, now supposing you have done, say, 30% ammonium sulfate precipitation before, but you want to increase it to say 60% ammonium sulfate precipitation. Are we following? You want to increase now to know what happens at 50%. Okay, some people, what they actually do is that they will get say 100 mils of the salt, the protein. First of all, they will add 10%, then 5.6 grams. Stir. After stirring for some time, do you understand? They will pour out about 10 mils from it and keep it as their 10% ammonium sulfate. So when they want to do 15%, they don't add 8.45. What they now do is that they will add an additional 2.8 grams. Okay, because the first one they already have 10%, so they will add 2.8 grams of ammonium sulfate, and then they will stir it maybe for 30 minutes. After stirring for 30 minutes in very cold buffer, they will pour out. Are we following? They will pour out. So when they pour out that they want to get to 20%, or because they already have 15, I wish there's a mark, there's a pen that I can use to color this. I wish there is something I can use to color this, so that, but there is none on the system. There is none on the system. Wow. Fortunately, there is none on the system. So if you have 20%, you want to achieve 25%, it means you're going to have add 2.7. So with that, you can get your different methods of protein precipitation. Okay, so after your ammonium sulfate, the best thing for you to do now is to remove the ammonium sulfate. And when we want to remove ammonium sulfate, we make use of dialysis. Okay, a dialysis, which you make use of dialysis bag. The dialysis bag contains pores which will allow the ammonium sulfate to move through the pores, but the protein will not move. Okay, so we put the protein in the dialysis bag and dip them inside a buffer. Okay, the same buffer that you have been using for extraction, and then you put it on a stirrer and you begin to stir. Sometimes you can stir for 4 hours, 5 hours, 24 hours. In fact, when I was doing an experiment with some proteins, I stirred from 6 in the evening till 6 in the morning. Okay, that is almost 12 hours to be sure that we remove all ammonium sulfate. Another thing you can do in removing it is using gel filtration. Since we know that the particles of the ammonium sulfate are quite small, smaller than our proteins, we can use a gel filtration to filter. And when we pour the mixture inside the column, the ammonium sulfate that is smaller will go inside the beads. Okay, and then the protein that is not small will come out. So we collect the protein first before we collect the ammonium sulfate. Okay, now when we remove ammonium sulfate, what we have actually is still known as our crude proteins. Okay, which means we still need to purify these proteins. We still need to purify these proteins. So I don't want this to be too bulky, so I'm going to stop here and then look at it in another video, the continuation of how we purify